Hello, welcome to STEM Andy. I will be presenting this episode myself, and the topic of choice this week will be on artificial nerve cells. I found a couple of interesting articles that I'll go over in a few minutes. But first, artificial nerve cells, also known as artificial neurons, are electrical or biological constructs that perform functions like real neurons. They are a rapidly growing area of organic electronic research, with potential applications in a variety of fields, such as bioelectronics, neuroprosthetics, and drug discovery. The ability of artificial nerve cells to mimic the electrical and chemical signaling of real neurons is one of their main advantages. This enables them to be used as models for studying nervous system function and dysfunction, as well as develop new therapies for neurological disorders. For example, by mimicking the electrical and chemical signaling of diseased neurons, artificial neurons can be used to study the underlying causes of neurodegenerative diseases like Alzheimer's, Parkinson's, and Huntington's. Neurons communicate richly to perform real-time cognitive computing tasks. Implementing these capabilities with electronic circuits is extremely difficult, and neuromodulation is neglected in neuromorphic circuits that mimic biological neurons. So understanding neuron processing and interpretation of neurons requires modulation. Based on its role, each neuron may have a unique modulation function. Before going into some of the articles I found, I'd like to dedicate some time to explain the difference between biological and artificial nerve cells. Biological nerve cells, also called neurons, are cells that are a part of a living thing's nervous system and oversee sending chemicals and electrical signals. They have a cell body, axon, and dendrites, and synapses are specialized structures that allow biological nerve cells to communicate with the other neurons. Artificial nerve cells, also called artificial neurons, are made of electronics or living things that act like real neurons. They are made to look like the electrical and chemical signals that real neurons send and receive. They can be used as models to study how the nervous system works and how it goes wrong, as well as to find new ways to treat neurological disorders. Real nerve cells differ in complexity, as they are complex structures with many shapes and sizes that process information in many ways. They communicate via chemical and electrical signals. However, artificial nerve cells are easier to make, but may not be as effective as real neurons, well, currently in their stage right now. Another distinction is their construction. Transistors, diodes, stem cells, and genetic engineering create artificial nerve cells. The cells divide and become nerve cells, and an important use for these artificial cells is in bioelectronics, where it can be used to create devices that communicate with the nervous system. The artificial neurons can be used to make neuroprosthetics that are devices, like I said before, to make neuroprosthetics which supplement function or the function of damaged or lost neurons. Cochlear implants for hearing loss, retinal implants for vision loss, and deep brain stimulations for neurological diseases. The living nerve cells can adapt to new experiences and learning. The artificial nerve cells are usually static and can't really change their behaviour, but they are very valuable in the drug discovery and the applications in things like medicine. The artificial neurons mimic the chemical signaling, can increase the discovery process to find effective drugs in neurological disorders. Despite these differences, artificial nerve cells may help researchers understand the nervous system and develop new treatments for neurological disorders. They allow controlled experiment-based study of the nervous system and its problems, and they can find that in this method will be used for creating the new drugs for the disorder treatments. In the 21st of April 2022, an article from New Scientist titled "Artificial Nerve Cells Have Been Made in the Lab" by Jason Miragesu was created. This is one of the earliest articles I could find that highlight the first implemented artificial neurons being made from hydrogel and it being 700 times wider than a regular neuron. The synthetic material was first implemented by Hagen Bailey at Oxford University using biocompatible materials, and this is first implemented as a stepping stone towards sophisticated artificial tissue to repair damaged organs. The ability to replicate the complex structure and function of real neurons is one of the most difficult challenges in the development of artificial nerve cells. Real neurons can come in a variety of shapes and process information in a variety of ways, so scientists are currently investigating a variety of approaches to try to understand and replicate the complexity of natural neurons. Now I'd like to go over the main article that influenced today's topic, and I took it from Science Daily. Since my recording, it was published 3 to 4 days ago on January 12, 2023. The article was titled "Artificial Nerve Cells Almost Like Biological." This was taken from Swedish researchers at Linköping University. They developed an artificial organic neuron that mimics biological nerve cells by sending natural nerve stimulation. And I wanted to highlight this in the opening episode as an example of emerging medical treatment and engineering. This promising method was developed at the Laboratory of Organic Electronics to enhance the artificial nerve cell models. They demonstrated the artificial organic neuron model and its approach to controlling a carnivorous ant's mouth, and it displayed 20 out of 20 characteristics that would distinguish it from a biological or a natural neuron. In their latest study, published by Nature Materials, LIU researchers developed a new artificial nerve cells called conductance-based organic electrochemical neuron, and to abbreviate that to COECN, that mimics 15 of the 20 neural features of biological nerve cells, making it more like natural neurons. A quote I took from the new study states that ion modulation is crucial to creating artificial neurons that mimic biological neurons, and silicon artificial neurons mimic many features but cannot communicate via ions. The COCNs demonstrate several biological neuron features, including ions, and from this I took that they were using silicon instead of the hydrogel, at least for this experiment. And the research group developed a N-type conducting polymer-based organic electrochemical transistor in 2018 for printable complementary organic electrochemical circuits. Since then, the research group has optimized the transistors for a better printing method on a flexible substrate to synthesize artificial neurons. Ions control electronic current flow through an N-type conducting polymer, causing voltage spikes. This mimics nerve cell activity in the artificial neurons. These artificial neurons have unique material that allows the current to be increased and decreased to mimic biological sodium-ion channels. The new COCN neurons were connected to the mice vagus nerves from the Karolinska experiments. The artificial neurons stimulated the mice nerves, causing a 4.5 heart rate change. This increased heart rate shows that the anti-ambipolar element is responsive to the ions and biomolecules, demonstrating that the neuron can be operated as an event-based sensor transition the biochemical signals to activate the vagus nerve of a mouse, showing the potential of the COCN closed loop regulation of the mice physiology. The artificial neuron has the ability to stimulate the vagus nerve and could lead to important medical applications. Organic semiconductors are compatible, malleable, they are soft, while the vagus nerve controls the immune system and metabolism. This is really important as the vagus nerve stimulation was used in the experiment as a therapeutic intervention for treating the epileptic seizures by controlling it and treating depression. And in the clinical trials for treating chronic inflammatory diseases like inflammatory bowel disease and rheumatoid arthritis, to further illustrate the possibility of using CLCNs in sensing nerve activation and biointegration, there was also a event-based sensing of glutamine and sodium at the neuron combined with stimulation of the right cervical vagus nerve, controlling the heart rate. The salt concentration can substantially shift the peak current voltage of the curve to modulate the spiking of the sodium OACT. The aim of doing all this was to reduce the energy consumption of the artificial neurons, which is considerably higher than that of human nerve cells, and the replication ability of this still requires much work. But even then, the paper still tries to introduce a new method to convert the continuous stimulus that shapes into modulated spiking and bursting patterns for these rich behavioural neurons. It used a Echevik mathematical model to support the excitatory spiking patterns and use Cadence virtual simulator tools to verify the signal the circuits implemented in a 45 nanometer complementary metal oxide semiconductor tech. And looking into biointegrated neuromorphic systems promised biological signaling recording and regulation protocols. For the artificial neurons needed a minimal complexity and iron-based operating mechanisms in biology. Previous models for the neuromorphic circuits for the biomimetic real-time cortex showed abilities like using the spike time dependent plasticity to mimic the learning patterns of biological neurons without forgetting and using dopamine signaling as a reward for the learning behaviour, and the spiking with a certain set of inputs, and most recently the spiking in the response of specific frequencies were used. The paper used certain tools to measure how effective the artificial neurons were. First, the OCTs, or the organic electrochemical transistors, from the mixed ion electron conducting polymers. That's basically just for the spikes and the process information using the action potentials, and specifically the spikes in the membrane potential. It also measured CMEM, which is known as the Comprehensive Modal Emission Model, using a variety of modal operations. This was optional in the circuit and can be embedded in the intrinsic capacitance of the OACT. And since the CMEM is the membrane capacitance, it also had the VMEM, which is the virtual memory, and was used as the membrane voltage. So they used different threshold voltages for mobility on the low and high voltage sides of the anti-amipolar transfer curve of the sodium OCT, representing the single and multiply charged species. It used Cadence virtual similar tools to show how the circuit works, and the modelling hypothesis says that the effect of stimulus dynamics of the action potentials may be caused by a type of neuromodulation that we don't fully understand yet. And one part about the structure of neurons is the area between the axon hillock and the soma, being called the axon initial segment. And recent studies shown that the axon initial segment, known as the AIS, may not just make action potentials, but process signals in real time, as this can be used as a processing unit inside the neuron and turns somatodendritic oscillations into action potentials. The neuromorphic implementation of the proposed model is a change to a previous understanding when using a voltage-based axon hillock design. And now this method in their paper shows that a direct link from the stimulus to action potential modulation. The COCN circuit used two OECTs, one sodium-based and one potassium-based device. The peak current voltage can be controlled using by tuning the ion concentrations in the electrolyte, and for a given concentration the different ions shift from a transfer curve by varying degrees. An example of these ions are calcium, magnesium and ammonium, showing lower peak current voltage than potassium and sodium. And the COCN action potential shows the typical features of the biological action potential, including the repolarization, depolarization, and hyperpolarization stages. So this is just some of the main features from this being the most recent paper on artificial nerve cells. As we can see, the biorealistic frequencies can be achieved with a neuron based on the three OCTs. They use different classes, class 1, 2, and 3 for the spiking, and they incorporated different additional channels in the circuit. Well, they used different additional channels in the circuit, like calcium modulated channels, which could enable possibly other missing features. And the COCN can be controlled with the secondary calcium ions and neurotransmitters, as that can also affect the peak current voltage. So to summarise the whole episode so far, this was just stuff I read on artificial nerve cells being a rapidly expanding area of biochemistry research. They have potential in applications in a wide range of fields. They enable the study of nervous systems, such as function dysfunction, as well as the development of new therapies for neurological disorders, and are extremely valuable as a tool in drug discovery. While replicating the complex structure and function of real neurons is difficult, we should still continue development of these cells, as it will greatly benefit many areas of science and technology. While artificial nerve cells are still in their infancy, they hold great promise for future understanding of the nervous system and developing the treatments. And that's basically all I've read up so far, and that's all I'd really like to share. As you can see, they're very promising and can really advance many areas like synapse formation, neuroplasticity, neurodegenerative disease, synaptic transmission regeneration, neural stem cells, glial cells, the information processing of nerve cells, neuronal signaling, and of course bioelectronics, which we've just gone over today. And it can even progress on areas that I might not have even mentioned. Even though it's still in early development, the impact and importance of this research on neuromorphic engineering is evident. Thank you for listening to my first independently directed episode. I'm planning to arrange an interview, but this could take a while because I'm currently completing my dissertation. So are many of my peers. It's not confirmed, but the next topic might be engineering based like today, depending on my availability to interview someone, and if not, it might just be another episode like this. For this podcast, I didn't mention it in the introduction episode, but I'd like to aim for around every second week, although it could be weekly depending on circumstance. Again, thank you for listening, and I hope to see you in future discussions.